Section 1. Seeing the Unseen. A world beyond our eyes. Microscopes are amazing tools. They are like windows to a world we can't see with our eyes alone. Think about it. There are tiny living things all around us. In a drop of pond water, on the back of your hand, even inside your own body. Without microscopes, we'd never know they exist. These instruments make tiny things look bigger. This process is called magnification. They help us see the incredible details of cells, bacteria, the building blocks of materials. It's a universe of the very, very small. Microscopes are our ticket to explore. This exploration helps us understand life, fight diseases, build new technologies. Microscopy is vast, but it boils down to two main types. First, the light microscope, the one you saw in a science classroom. It uses visible light to magnify. Second, the electron microscope, a much more powerful, complex machine. It uses a beam of tiny particles, electrons. They work very differently, so they show different things and serve different jobs. Section 2. The Light Fantastic. How light microscopes work. Let's start with the classic, the light microscope. Its name gives you a big clue about how it works. It uses light. Specifically, it uses visible light, the same stuff that comes from the sun, or a light bulb. Diagram. Path of light through a compound light microscope. The setup is elegantly simple. There's a light source at the bottom which shines up through your sample. The sample itself has to be thin enough for light to pass through it. Above the sample, there are a series of glass lenses. These lenses are curved in a special way. As light passes through them, they bend it, spreading it out. This bending of light is what makes the image of the sample appear much larger. You look through a lens at the top called the eyepiece. The final image you see is magnified twice. First by the objective lens, which is the one right above the sample, and then again by the eyepiece lens. If the objective lens magnifies the image 40 times and the eyepiece magnifies it 10 times, your total magnification is 40 times 10, which is 400 times. Most standard light microscopes can magnify things up to about 1,000 or maybe 1,500 times. But there is a limit. This limit is not because of the quality of the glass, but because of the nature of light itself. Light travels in waves, and you can't use it to see things that are smaller than its own wavelength. This limit is called the resolution limit. Resolution is all about clarity. It's the ability to tell two very close objects apart. Think of it like the pixels on your TV screen. The more pixels, the sharper the picture. The wavelength of visible light is between about 400 and 700 nanometers. Because of this, a light microscope can't distinguish between 2 points that are closer than about 200 nanometers. If two tiny dots are only 100 nanometers apart, a light microscope will just show them as one blurry dot. This means even the best light microscope just can't see ultra-tiny things like a single virus or a ribosome inside a cell. One of the greatest things about light microscopy is that you can look at things that are alive. You can place living cells in a drop of water on a glass slide and watch them in real time. You can see them move, eat, and even divide into new cells. This is incredibly powerful for biology. To make certain parts of the cell easier to see, scientists often use stains. These are special dyes that stick to specific structures, like the cell's nucleus or its outer wall, making them stand out with color. This helps us identify different parts of the cell and understand what they do inside a living organism. Section 3. Under the beam, the power of electron microscopes. Now, what if you need to see things that are smaller than 200 nanometers? What if you want to see the intricate details of a virus or the fine structures inside a cell's powerhouse, the mitochondrion? For that, you need a different kind of tool, an electron microscope. This machine doesn't use light at all. Instead, it uses a beam of electrons. Electrons are super tiny, negatively charged particles. Their wavelength is thousands of times shorter than visible light. That shorter wavelength is the secret to the electron microscope's power. It can see things in much greater detail. Because it uses electrons, an electron microscope is built very differently. It doesn't use glass lenses. Instead, it uses powerful electromagnets as lenses. Electrons are scattered by air molecules, so the whole path from source to detector is kept in a near-perfect vacuum. This is a big, complex, expensive setup. The final image isn't viewed through an eyepiece. It's formed on a fluorescent screen or captured by a digital camera and shown on a computer monitor. There are two main types of electron microscopes. The first is the transmission electron microscope, or TEM. In a TEM, the electron beam passes through an extremely thin slice of specimen. Electron gun, magnetic lenses, specimen, final image on a screen. It's like a superpowered version of a light microscope. Some electrons are scattered by dense parts while others pass through. The resulting image shows internal details. A TEM can reveal tiny organelles like ribosomes, endoplasmic reticulum. It can magnify up to 1,000,000 times or more, with resolution better than 0.1 nanometers. The second type is the scanning electron microscope, or SEM. An SEM is like feeling an object with your fingertips in the dark. It scans a focused electron beam across the surface. The beam knocks other electrons loose. Those electrons are collected by a detector. A computer builds a 3D-like image from the signals. SEM shows surface texture, like the intricate pattern on a fly's eye, the structure of a pollen grain. Magnification is lower than TEM, up to about 500,000x. Section 4. Seeing is believing, a tale of two images. The most obvious difference is the image itself. When you look through a light microscope, you can often see color, natural color, like green chlorophyll in a plant cell, or color from stains scientists add to highlight parts. Stains make images vibrant and easy to read. Nucleus stained deep purple. Cytoplasm stained light pink, a world of color. In stark contrast, electron microscope images are black and white. Because the microscope detects electrons, not light, the image maps electrons that pass through the sample in a TEM, or that were scattered from the surface in an SM. Darker areas, chakras, more electrons blocked, chakras, denser structures. Lighter areas, chakras, more electrons detected. Scientists often add color later with software. That's false coloring, but the raw data is grayscale. You can't look at living things with an electron microscope. EM sample prep is harsh, fixed with powerful chemicals, completely dehydrated. For TEM, sliced into ultra-thin sections. That process kills the cell. So EM gives a static snapshot, a dead cell frozen in time. You get incredible detail, but lose the dynamic story of life. A light microscope is best for watching life unfold, like bacteria swimming. Practical example, a human red blood cell. Under a good light microscope, you can see it clearly. A small, reddish, donut-shaped disc. You can see its shape and size, and watch it move in a live sample. Under an SEM, you see the surface in 3D detail. The smooth, concave curve becomes visible. With TEM, you could slice through it. No nucleus or mitochondria, just hemoglobin. Each microscope tells a different, equally important part of the story. Section 5. A brief history of looking closer. The story of the microscope didn't begin with a flash of genius, but with centuries of curiosity and tinkering. The journey to see the small stuff really got going in the late 1500s. Around 1590, Hans Janssen, Zacharias Janssen. They are credited with putting lenses together in a tube to make the first compound microscope. It was a simple device, but it planted a seed. The real wow moment came later. In 1665, Robert Hooke used his homemade microscope to look at a thin slice of cork. He saw countless tiny, empty boxes that reminded him of the small rooms in a monastery. He called them cells, a name that stuck forever. Just a few years later, another pioneer took microscopy to a whole new level, Antony van Leeuwenhoek. A Dutch draper, not a trained scientist, but a master lens grinder. He crafted tiny, powerful single-lens microscopes, more like powerful magnifying glasses. He became the first person to see and describe bacteria. He called them animalcules, little animals. He saw them wiggling around in a drop of water. He also observed blood cells, sperm cells, and much more. Van Leeuwenhoek opened up an entire microbial world, laying the foundation for microbiology. For the next couple of hundred years, light microscopes got better and better. Lens makers learned to correct distortions and improve image quality, but they always ran into the fundamental wall, the wavelength of light. Scientists suspected structures smaller than what light could show. The big breakthrough came in the 1930s. Physicists realized electrons behave like waves. In 1931, two German engineers built the first transmission electron microscope, Ernst Ruska, Max Knoll. It was a revolutionary moment. This invention opened the floodgates. Scientists could finally peer inside cells and see internal machinery in exquisite detail. Ernst Ruska later won the Nobel Prize in Physics in 1986. The scanning electron microscope followed. Early versions in the late 1930s, commercial availability in the 1960s. Scientists could now see surfaces in stunning three-dimensional detail. The electron microscope didn't replace the light microscope. It complemented it, launching science into the nanoworld. Section 6. Pushing the limits. Modern microscopy marvels. The world of microscopy is not standing still. Far from it. Scientists and engineers are constantly inventing new ways to see smaller, faster, more clearly. One of the most exciting areas of advancement has been in light microscopy. For a long time, everyone accepted the 200 nanometer resolution limit as an unbreakable rule. But in recent years, several clever techniques, collectively known as super-resolution microscopy, have smashed right through that barrier. Methods with cool names like STED, Palm, Storm use fluorescent molecules and computer tricks to achieve resolutions as sharp as 20 nanometers. This allows scientists to watch molecules interacting inside living cells, bridging the gap between conventional light and electron microscopy. On the electron microscope side, a technique called cryo-electron microscopy, or cryo-EM, has caused a revolution in biology. Instead of using harsh chemicals and embedding samples in resin, cryo-EM involves flash freezing the biological sample in a thin layer of ice. This freezing happens so fast that ice crystals don't have time to form, preserving the sample in a near-native state. The TEM is then used to take thousands of pictures of the frozen molecules from different angles. A powerful computer then combines all these two-dimensional images to reconstruct a highly detailed three-dimensional model. This technique has become so important that its developers were awarded the Nobel Prize in Chemistry in 2017. The digital revolution has also completely transformed microscopy. Gone are the days of squinting through an eyepiece and sketching what you see. Today, high-resolution digital cameras are standard on almost all research-grade microscopes. This allows for easy image capture, storage, and analysis. Powerful software can stitch together thousands of images to create a huge, detailed map of an entire tissue sample. Furthermore, artificial intelligence is now being used to analyze these massive data sets, automatically identifying cells, spotting signs of disease, or tracking particles as they move. This automation speeds up research immensely and reduces human error. Another exciting trend is making microscopy more accessible. While high-end electron microscopes are still massive, expensive machines confined to special labs, light microscopy is becoming more portable and affordable. There are now high-quality portable microscopes that can be used for disease diagnosis in remote villages. There are even attachments that can turn a regular smartphone into a surprisingly powerful microscope. These innovations are putting the power of seeing the unseen into more hands than ever before, democratizing science and enabling new discoveries in classrooms, clinics, communities around the world. These modern marvels continue to expand our vision, promising even more incredible discoveries to come. Section 7. The right tool for the job. Making the choice. So, after this journey into the world of the very small, we come back to our original question: which microscope should you use? The answer is simple. It all depends on what you want to see. There is no single best microscope. There is only the best microscope for a specific task. Think of it like a toolbox. You wouldn't use a sledgehammer to hang a small picture frame. You wouldn't use a tiny screwdriver to break up concrete. Light microscopes and electron microscopes are different tools for different jobs. Each has its own unique set of strengths and weaknesses. To summarize, choose a light microscope when your priority is looking at living cells and processes. If you want to watch a cell divide, see bacteria swim, observe how a tissue responds to a drug in real time, the light microscope is your only choice. It's also the right tool for routine tasks that require speed and simplicity, like checking a blood sample in a hospital teaching students about cell structure in a biology class. They are relatively inexpensive, easy to use. Sample preparation is quick and straightforward. Their limitation is resolution. They can't show the finest details. Turn to an electron microscope when absolute top priority is detail. If you need to see the precise structure of a virus, visualize the folds inside a mitochondrion, examine the atomic lattice of a new material, you need electron microscope resolution. The trade-off: these machines are expensive, require highly trained operators, involve complex, lengthy sample prep that kills the specimen. You get a static black and white image of unparalleled detail, but you lose the dynamic context of life. In the end, light and electron microscopes are partners in discovery. Scientists often use both to get a complete picture. Use a light microscope to find an interesting event in a living cell. Then use an electron microscope to zoom in on the ultrafine structures, from Hooke's first glimpse of cells to the latest cryo-EM models of proteins. Microscopes have expanded the frontiers of human knowledge. By understanding what each tool does best, we can continue exploring the intricate, invisible world that surrounds and defines us.